rounds. So this morning, it's our pleasure. We have our own Dr. Kevin Burns going to be speaking to us today about, I think, some work from his lab, and specifically on MicroRNA Therapeutics in AKI, and looking at some of the preclinical evidence. So I'll hand the platform over to Kevin.
All right, thanks, Greg. I picked this title really to let everybody know that this, talk's a little bit different from your usual nephrology grand rounds, and that I'm going to be focusing on work, laboratory-based work related to AKI, and work we've been doing over the past, I would say, 2 to 3 years in this topic. So I'm not going to talk really about clinical data or patient-related data today. And I have no disclosures. Just outline what I'm going to be talking about. So I'd like to just review the topic of what are microRNAs and their involvement briefly in kidney disease, and then talk about the role of microRNAs in acute kidney injury. And then the last part of the talk, I'm really going to focus on the experimental work we've been doing in the lab with a particular microRNA called 486.5P. And we've been studying it in a mouse model of ischemia reperfusion AKI, but also we've conducted recent studies to look at the effects of this microRNA on the genes which are expressed in the kidney. So you've all learned probably way back in either high school or university days about the transcriptome, really, and these are the RNA families in the mammalian transcriptome, but this subject has evolved over time. There are roughly 30,000 genes in the mammalian transcriptome, but surprisingly only about 2% of these are actually coding for messenger RNA, which is the topic you probably are most familiar with. And the messenger RNA, of course, gets translated into proteins. And then I guess before the 1990s, it was unclear what the role of the remaining 98% of these non-coding RNAs was. And a lot of people thought, well, maybe it's just junk RNA that's doing nothing. But of course, that's not true. So these non-coding RNAs are now thought to be very important. And they're basically subdivided into 2 groups. The long non-coding RNAs are more than about 200 nucleotides each. And one of the categories here is called long non-coding RNAs. I'm not going to talk about that today, but there are several very good reviews on this topic, even in the kidney literature recently. And these RNAs, they bind to histone complexes on the genome, and they can affect transcription and are heavily implicated in as having important roles in AKI and CKD, in transplant function, and in renal fibrosis. And then the other category are the RNAs which are less than 200 nucleotide length. And there are various categories, and some are found primarily in the nucleus, but some are found also in the cytoplasm. And they encompass a wide variety of groups: silencing RNAs, small nucleolar RNAs, nuclear RNAs, and the topic today that we're going to focus on is what's called microRNA. And all of these categories really are of RNAs are important because they regulate the expression of genes and the translation to protein. So what are microRNAs? Well, the first microRNA was discovered in 1993 in the nematode C. elegans. It was called Lin4. And that discovery was, in those experiments at the time, it was shown that this particular microRNA had a major role in affecting the function of the nematode and neurologic functions. And subsequently, the 2 individuals who did those experiments were awarded the Nobel Prize in 2006. So that tells you something already of the importance of this field. MicroRNAs are 18 to 25 base pairs in length. And since 1993, over 2,000 mature microRNAs have been identified in humans, and they're thought to comprise about 1% of all human genes. And there's a website that has the entire catalog of these microRNAs. They're highly conserved across species. So, you know, the structure of a microRNA found in mouse, for example, is usually identical to the one that's found in humans. And they control about 50% of the mammalian protein coding genes. And also they're differentially expressed in human tissues. What I mean by that is that some are particularly highly expressed in the kidney, but not in other organs. Interestingly, they can be found in plasma and urine and other body fluids. They're fairly stable. And why is that? Because they're packaged, and they're packaged in a protein complex. The protein complex is called argonaute, and that protects them from endonucleases, really. So because of that feature, a lot of the research on microRNAs has been focused on their biomarker potential in various diseases, particularly cancers, myocardial infarction, but also kidney disease. So how are they synthesized and what do they do? This is just a diagram of a cell with the nucleus circled. So they're transcribed by RNA polymerase II here as genes. And then the RNase polymerase II will transcribe what's called a primary microRNA, which is usually about 70 base pairs or so, and it has a hairpin structure. And then within the nucleus, there's an enzyme called DROSHA that cleaves this primary microRNA into what's called a pre-microRNA. And then through this protein called exportin 5, the pre-microRNA is exported out into the cytoplasm as this hairpin RNA structure. And then it's acted upon by an enzyme called dicer. And dicer then cuts the tip of the hairpin loop and creates this microRNA duplex structure. And so two strands that are complementary to each other. And then one of the strands will be so-called lead strand is directed or targeted into something called the RNA-induced silencing complex, or the RISC. And then once it ends up in that complex, it gets directed to the target messenger RNA. And so there's usually a sequence on the microRNA, and might be about 6 to 8 nucleotides that will bind to a target sequence on messenger RNA at the 3' untranslated end. And when this microRNA binds to that target sequence, it stimulates, or it leads to destabilization and degradation of that messenger RNA. Now, when there's a 100% identity of the sequence to the 3' end, that usually leads to this degradation. But oftentimes, microRNAs, and this is an important point, they tend to be quite promiscuous. So any particular microRNA may be targeting several, up to hundreds of different genes, depending on the sequence. When there is not 100% sequence complementarity to the 3' end, that can still lead to binding, some binding, and repression of translation. So you can see here that they're really quite potently involved in regulation of gene expression. But this also leads to obviously challenges in therapeutics, because any particular microRNA may have significant off-target effects. This just to show you that it's from a recent review that's focused on some of the microRNAs that have been studied in CKD and both experimental models and human observational studies. And a lot of the research has focused on diabetic nephropathy. There are large numbers of microRNAs that have been studied. And one of the common microRNAs that affects both diabetic nephropathy as well as CKD-related cardiovascular disease, hypertension, and renal fibrosis is this one called MicroRNA 21,5P. And I'll talk a little bit more about this later, but this particular RNA appears to be anti-fibrotic, let's put it that way, so that it prevents the developments of long-term complications of CKD. And I've circled here this one called MIR4H65P, which I'll spend a lot more time talking about. This particular microRNA in CKD has been shown to be involved in the sarcopenia of CKD. So in animal models, the expression levels in muscle of this microRNA appear to be suppressed. And when those animals are transfected with this microRNA, that appears to have a beneficial effect on muscle function and muscle growth. And just to let you know that this is not just kind of all focused only on experimental studies and human observational studies. There are clinical trials of MicroRNA therapeutics in kidney disease, and I'm aware of at least 2 trials ongoing or completed. The upper part of the slide shows a trial sponsored by Genzyme that's looked at silencing microRNA 21 for the treatment of Alport syndrome in animal models. This has been shown to be an effective strategy to decrease proteinuria and glomerular injury. However, this study involved a small number of patients. It's a phase 1 study, and it finished, completed in May 2019, and I still haven't seen any publication related to this work. So that's a little bit concerning, and I don't know what the ultimate results of that trial are. And a second study involves Regulus Therapeutics, and this is looking at autosomal dominant polycystic kidney disease and a particular microRNA called microRNA 17. And in animal models of PKD, this microRNA appears to be involved in cyst growth. And so that delivering this anti-microRNA in animal models has been shown to suppress cyst growth and improve kidney function with time. This particular phase 1 trial is still ongoing as of April 2021. So what about microRNA in acute kidney injury? What do we know about this? The first study that looked at this or addressed this question was published more than 10 years ago in JASN by a group in the US, and it was quite an elegant study. And what they did here was they created a mouse model in which the enzyme that I talked about earlier called dicer was selectively deleted from proximal tubules using Crelox technology in these mice. And you can see on the left part of this slide that when they selectively knocked out this enzyme dicer that's responsible for maturing the microRNA, they were able to eliminate almost all of the microRNAs that they examined. And that's shown in green here. So the green signifies a low level of expression. The red signifies high level of expression. So this was a targeted approach. And then when that was done and when the animals were subjected to ischemia reperfusion injury, you can see that these animals were protected significantly from kidney injury as per the BUN and creatinine. And they also had diminished levels of apoptosis in the kidney. And apoptosis or programmed cell death is really a classic marker of tubular cell injury following ischemia reperfusion. So this study highlighted the importance globally of microRNAs in the pathogenesis of AKI, but did not specifically focus on a single category. Subsequently, there's been a lot of work looking at microRNAs in AKI and non-coding RNAs. MicroRNA 21 has been the one that's most widely studied. And it's one that inhibits apoptosis and inflammatory responses in the kidney. Its expression level is increased in the kidney, the heart, and the lungs. And that's why it's been a subject of a lot of interest. And then in human AKI, most of the studies that have focused in this area have been observational studies. I just highlighted a couple. There are 10 differentially expressed microRNAs that have been identified in serum of patients with AKI. compared to healthy controls, and a study in the NDT in 2016 showed that plasma levels of microRNA had predictive value for AKI after cardiac surgery and with an area under the curve of 0.7. Because there's really no treatments for AKI, as you know, this begs the question of what about the potential therapeutic role of microRNAs in this condition? So we were interested in trying to answer that question, and we began by doing a systematic review that focused on experimental studies in AKI. And we were primarily focused on manuscripts that used microRNA or delivered microRNA or antagonists of microRNA in animal models of AKI. So this involves all non-human studies with acute kidney injury. So we isolate, we found about 2,000 abstracts, and after screening of the abstracts, we ended up with 70 papers for the review. And this shows you some of the features of the study. So about 83% of the studies examined the effects of direct delivery of either a mimic of the microRNA, which is simply a identical sequence, or what's called an antagomer or locked nucleic acid derivative that would bind to the microRNA, inhibits its activity. And the remaining 17% or so of the studies involved delivery of microRNAs through cells that were highly expressing that microRNA, or extracellular vesicles, or actual deletion of genes involved in or producing that microRNA. We ended up finding 42 microRNAs that were studied in the 70 papers. And as expected, microRNA 21 was the most common microRNA evaluated, but most of the other studies were single studies. microRNA 146 and 155 were also more than 2 studies each. And this slide really shows you some of the, I guess, challenges of basic research in acute kidney injury. And we were not too surprised to find this, but you'll see that all of the studies involved rodents. So all of the 70 papers. And not surprisingly also, 79% of these studies involve male rodents. And there is a bias to use male rodents in AKI studies for the sake of simplicity and the sake of not having complicated, or the issues complicated by the estrus cycle. And some studies actually did not even report the sex of the animals. There are a limited number of models that have been used with microRNA. Most of the studies involve ischemia reperfusion injury. So typically this is done with either unilateral clamping of the renal pedicle or bilateral clamping followed by reperfusion. Sepsis models were the next most common, and this typically involves ileocecal ligation and then puncture of the ileocecum leading to sepsis. And a smaller number were related to nephrotoxicity, and most of these studies involve use of cisplatin. A small number of studies actually were studying rhabdomyolysis. Now, what were the biological processes? Well, I guess one of the first things I want to emphasize is that, and it's a really maybe a weakness, 100% of these studies showed a significant effect of the microRNA intervention, either positive or negative, on AKI. And that begs the question, of course, what happened to the ones where there were no effects? And obviously there's a publication bias. Now, of the biological processes that were studied, there were actually 17 identified biological processes affected by the microRNAs, and the top 3 processes were apoptosis, which I've talked briefly about, inflammatory responses in the kidney, and oxidative stress. And then the most common pathway linked to these processes was the so-called PI3 kinase AKT pathway. Now, PTEN is a tumor suppressor gene. It's ubiquitous, and it's involved in, when it's stimulated, leading to programmed cell death or apoptosis. And interventions that block PTEN tend to lead to cell proliferation or cell differentiation. And in the case of these studies focused in this area, inhibition of PTEN in 83% of the cases was protective against AKI. We went on to conduct a meta-analysis. There were 14 studies that focused on microRNA 21 in experimental AKI, and 12 of these studies had data on serum creatinine or kidney injury scores to allow for a meta-analysis. And that's shown on this slide. The studies were divided into two groups. So there were 9 studies that involved preconditioning and then use of a microRNA 21 antagonist. And preconditioning means an intervention that actually supposedly will protect the kidney from subsequent injury by the intervention. So this involves ischemic preconditioning or use of xenon gas or cobalt chloride. And in those 9 studies that looked at the effect of the microRNA antagonist, there was a highly significant effect favoring the control. In other words, the antagonists tend to worsen the kidney injury as determined by serum creatinine here, or kidney injury scores with a p-value was quite low, although there was a very significant degree of heterogeneity in these studies. Now, in the absence of preconditioning, so this is where the antagonist is simply given without ischemia or xenon, et cetera, we found that there was no overall significant effect of the microRNA 21 antagonist on the outcomes. One of the caveats to mention about this is that all of the all of the preconditioning studies involved papers coming from one single group, even though the authors' names tended to change, or the first authors' names. It's all from one lab, and that's obviously a bit of a red flag in terms of how one can make strong conclusions about this. Overall, it looks like microRNA 21 may have some promise with respect to AKI, but obviously this has to be duplicated by other groups. And I'm not going to talk about some of the other studies that are quite interesting. 146 and 155 microRNAs also have potential in my mind. And then the other point to emphasize in this kind of a systematic review, looking at experimental studies, is that the study quality tends to be quite low. So we looked at the risk of bias for these studies, and in orange here you can see how we classified bias as unclear in most of the categories. And for example, many of the papers, if not most of the papers, did not specify whether the interventions were blinded or the outcome assessments were blinded, et cetera. There was missing data that was evident in many studies. And it's a common feature, unfortunately, with experimental studies, and it highlights the need for more rigorous study design and also use of other models of AKI that are more appropriate to the human condition, and also studies that should balance male and female sex. So I'll switch gears now and then talk to you about our area of interest over the past couple of years, and that's microRNA 486.5p. And I'll just start by showing you why we're interested in the proximal tubule as a particular target in AKI, and this is nothing new, I hope, to any of you. But the kidney, of course, is quite susceptible to ischemic or hypoxic injury And that region of the kidney that is most highly sensitive is the proximal straight tubule S3 segment. And that's because it sits in a relatively hypoxic environment, and it also utilizes high levels of ATP for transport. And the other point to show on this slide really is that the tubules are closely approximated to the vasa recta, so that endothelial cells are very close nearby. I'll show that on the next slide. And there's really crosstalk between the endothelium and the proximal tubule in the kidney. Now, the kidneys, of course, have the capacity to regenerate and recover from AKI. And on the upper part of the slide, you'll see a normal tubule subjected to injury. There's apoptotic cells that slough off into the lumen. However, with a normal regenerative process, one can then completely restore kidney function. And many of our patients, of course, are admitted to hospital and get discharged with normal serum creatinine or back to their baseline serum creatinine after AKI. However, on the bottom part of the slide, you'll see that the tubule sits very close to the renal capillaries and vasa recta. And during ischemia, for example, the endothelial cell will become quite permeable and will lead to inflammatory cell infiltrate and cytokine production within the interstitium that can cause or propagate tubular cell injury. And when this happens to an extensive degree, then you have this maladaptive recovery response shown in the middle part of the slide, where there's failed differentiation, and this leads to CKD or renal fibrosis, or the so-called AKI to CKD transition. Because of the ability of tubular cells in the kidney to regenerate, there's been a lot of interest in the use of stem cells or their products in trying to accelerate recovery from AKI. And we've been interested in this area for about 10 years now. And I first got interested in collaboration with Dave Allen in the Division of Hematology. And he was isolating cells from human cord blood that in other experimental models were found to be quite beneficial in terms of reparative functions. And so we studied this, we studied one of these cell types. It was called CD133 positive cord blood cells. And our initial studies actually showed that administering these cells to mice with AKI actually worsened the injury. So we were a bit discouraged, but about 6 years ago now, Dylan Berger, who was in my lab at the time, led some studies in which we examined another type of cord blood cell called endothelial colony forming cells, or ECFCs. And in the studies that he led at the time, injecting these cells into mice with ischemia, reperfusion injury, we were able to show that this had a potent reparative effect on kidneys after ischemia. However, we found that these cells, when they're injected into the mice, did not appear within the kidneys or any other organs, and we looked at various time points. And so at the time, there were a number of papers coming out showing that in the field of stem cell biology, that some stem cells were releasing small vesicles called exosomes. And these are 50 to 100 nanometer diameter vesicles shown in diagrammatically here. And they're packaged with cargo, and the cargo contains microRNA. They're released from cells, and then they travel to the target cell, in this case, the injured cell within the kidney, where they deposit microRNA or messenger RNA, et cetera, which then leads to actions. So we wondered whether these exosomes could be playing a role. And in the studies conducted by Dylan, he showed that the exosomes actually also had the same effect as the cell delivery itself in protecting these mice from kidney injury after ischemia. So the next question was, well, what is it about these exosomes that causes repair or promotes repair? And so we conducted small RNA sequencing of the contents of these exosomes, and we ranked all of the microRNAs that were isolated from these ECFC-derived exosomes. And lo and behold, this particular microRNA called 486.5p was shown to be highly enriched in these exosomes at a level almost 300-fold increase compared to another type of vesicle released by the cells, and also about 30-fold higher than the cell itself. And the next highest ranked microRNA, which was 25.3p, was only 4-fold increase. So this was a very unusual enrichment of this particular RNA, microRNA, and it led to the question of, well, is it a candidate for the therapeutic effects that we were seeing? So we went on to do some further experiments focused on answering that question, and this is just one of them. We had mice with ischemia reperfusion AKI, and we injected them with exosomes at the time of ischemia reperfusion injury. And as you can see in this slide showing creatinine and BUN, 24 hours after ischemia reperfusion, there's an increase in creatinine as expected. This is as expected, but also remarkably, I think, we saw that the exosomes completely blocked this increase in creatinine and BUN. However, when we took the exosomes and pre-incubated them with an antagomer to this microRNA 486.5p, this prevented the preventive or block the ability of these exosomes to prevent injury. So that suggested to us that this microRNA might indeed be the candidate that's inducing repair. So the next series of studies that we've been conducting is really to determine if we could deliver this microRNA specifically to mice with a kidney ischemic injury. And we were also interested to determine what is the effect of this microRNA on the transcriptome in these cells of interest, the proximal tubule cell and the endothelium. There are a number of challenges to delivering microRNA for therapeutics. If one delivers microRNA in vivo, it is quickly degraded by endonucleases, and therefore there are various strategies that have been developed to prevent that. And one of them is to use pegylated nanoparticles shown here in yellow, or one can use cationic liposomes as shown in the middle, which kind of trap the microRNA and protect it from being degraded. And then more recently, there's been a lot of interest in using these either viral vectors or liposomal products or nanoparticles that are coated on their surfaces with targeting proteins that can direct these therapeutic agents to particular cells in the organ of interest, for example. And specifically, this has been applied to therapeutics in cancer. So in our case, we elected to package our microRNA in a commercial product. It's called in vivo fectamine. It's a liposome-based derivative, and it's been shown in a number of studies to be highly effective in allowing delivery of microRNA to distant organs. Before talking about the results, I just want you to know that we did look at the possible differences between male and female mice in our model. And this has been the subject of a number of other groups as well. And highlighted on this slide, you'll see the serum creatinine in this model of ischemia reperfusion injury. And you'll see that males, are much more sensitive to ischemia reperfusion than female mice, although you can argue, of course, that this is with serum creatinine and female mice may have lower muscle mass. That's true, certainly true. So creatinine is not the best marker. But when we look at other injury markers like NGAL shown at the lower part of the slide here, you see that the mice, female mice are protected. And I show this heat map here on the right. And these are genes in the proximal tubule and the endothelium using this RNA sequencing platform that we have studied. And you can see here that with ischemia reperfusion in the male on the far right, there are numerous genes either activated or decreased in expression following ischemia reperfusion compared to the sham male mice. But quite remarkably, in the female mice, it doesn't look like there's very much going on at all with ischemia reperfusion, both in the proximal tubule and the endothelium. So I'm not going to talk more about what that signifies, but it's an area of particular interest in our lab as well, sort of categorizing the genes that are potentially protective in females and the mechanisms for that protection. So that was my, I guess I would say my full disclosure slide, because this slide shows the protocol that we've been doing for our studies with microRNA 486.5p, and we've been studying male mice for the reason that they are more susceptible to ischemic injury. And so the mice are subjected to either sham surgery or 30 minutes of bilateral renal pedicle clamp, followed by release of the clamp. And at the time of release of the clamps, they're injected intravenously through the tail vein with either vehicle, a mimic of the microRNA 486.5p, or a control called scramble microRNA. And so this is a sequence that's just a scramble of nucleotides, not expected to induce any effects on gene expression necessarily. And then as our positive control, we have these ECFC-derived exosomes, which are, as I've said, enriched in this microRNA. 24 hours later, the animals are sacrificed. We take plasma for assessment of renal function, and we isolate kidneys as well as other organs. And we've been isolating endothelial cells from these kidneys, extracting RNA as well as proximal tubules. And we conduct real-time PCR or RNA sequencing, library creation, and then bioinformatics on the genes. And we also, of course, study the kidney sections and homogenates and do a number of things, including real-time PCR, immunoblots, apoptosis studies, and I'll show this in a second. So this first slide shows what happens to the microRNA once it's injected into the mice with renal ischemia reperfusion, and this is at 24 hours after injection. So we found that within the liver and the spleen that there were increases in this microRNA up to 24 hours after injection, and also in the kidneys. There was a small increase with the exosome injection, but it was not as significant as with the mimic itself. And of course, the scramble microRNA had no effect. And then when we isolated proximal tubules, we saw that these cells also had significant increases in microRNA 486.5p, as well as the endothelial cells. We also looked at the lung, the heart, and the brain, and there were no changes in the levels of microRNA 486.5p in those organs. This slide shows you in situ hybridization. So in situ hybridization just means taking a complementary probe for the microRNA of interest and labeling it for immunodetection on kidney slices. And we looked at this at 30 minutes after injection and at 24 hours after injection. And in graphic display here on the right, you can see that with the mimic and the exosomes, we saw higher levels of microRNA within cells after 30 minutes. And this persisted with use of the mimic, but not with exosomes at 24 hours. And just to highlight where we saw the microRNA within the kidney, That's on the bottom panel under IR plus mimic. You can see that it's in the cytoplasm of tubular cells, but interestingly, it was also heavily found within interstitial cells and endothelial cells that were in proximity to the tubules as well. So what was the effect of this injection? This is shown on this slide with the creatinine and urea, as well as kidney histology. So as per the previous slides, you can see that ischemia reperfusion after 24 hours, there's an increase in the serum creatinine as expected. An injection of the mimic completely prevented this and was similar to the effect we saw with exosome injection, whereas there was no impact of the scramble. While this was significant for creatinine, the results with urea were, there was some decrease with mimic, but this did not reach statistical significance. However, looking at injury scores, there was a clear effect, protective effect of the mimic on kidney histology. Now recall that this is injecting the mimic at the time of reperfusion, so at the onset of AKI. We went on to do a small number of studies to look at, or to try to answer the question, what happens if we inject this mimic after the onset of injury? And that's, I think, more clinically relevant because most of the time when we see patients, it's long after they've had renal injury. So we looked at a time point of 6 hours after ischemia reperfusion injury. That's when we injected the mimic in this case. And we were very surprised. There's quite a bit of variability in these results, but we were surprised to see that the mimic continued to have this protective effect on renal injury, even at that later time point. One of the markers of proximal tubule injury is megalin expression. So megalin is a protein expressed on the brush border of the proximal tubule cell. It's involved in the reabsorption of proteins and albumin, for example. And with ischemia reperfusion, we completely lose staining for megalin in the kidney, but both exosomes and mimic cause a partial recovery of this staining. And of course, here scramble has no impact at all. And then with regards to this phenomenon of apoptosis, the results were quite similar. We did two different assays for this. One is called a tunnel assay, which looks at fragments of DNA and stained in brown here with ischemia reperfusion. You can see most of the apoptosis occurs within tubular cells. Here on the right under B, you can see that endothelial cells also undergo apoptosis in this model. And the effect of the mimic is to cause a partial restoration of, or partial protection against apoptosis compared to ischemia reperfusion alone. And then we conducted another assay that's called caspase 3 activity, which is an enzyme that's triggered in apoptosis. And again, the mimic and the exosomes both prevent activation of this enzyme in the kidney. I'm not going to spend a lot of time on this, but just to highlight the fact that this tumor suppressor gene called P10 that I referred to earlier is also activated with ischemia reperfusion, and this is immunohistochemistry. And you can see that both exosomes and mimic suppress the expression of this protein within the kidney by immunohistochemistry. Now, we're not aware of any other studies that have performed immunohistochemistry for P10 in the kidney. And we were very surprised to find that P10, as it turns out, is highly expressed in the juxtaglomerular cells. And that's, of course, where renin is produced. We don't know the meaning of this, but you know, it's obviously a project worth waiting to be done. And if anyone's interested, please come talk to me, because it's not something that we necessarily will have time to do ourselves. Okay, so what happens to the genes in the kidney when we inject the microRNA 486 5P? This was the second most important thing that we wanted to study in our model. And this shows this slide shows a heat map for proximal tubular genes and endothelial genes. So with all of the conditions that we studied, and it just highlights the fact that with ischemia reperfusion in the proximal tubule, you see many genes that are in blue means downregulated, but many genes upregulated in red. And then you can see just by the pattern that both the mimic, there were 2 samples available for the mimic and 3 for the exosomes, but that there is a reversion of the pattern closer to the sham mouse. However, we were surprised and I guess initially disappointed to see that the scramble microRNA also had an impact on genes in the proximal tubule and in the endothelium. But it turns out that this ended up being helpful for us in doing the bioinformatics. So one way to look at the impact of the genes is to construct what are called volcano plots, in which one compares the gene expression pattern in 2 conditions. So here we're comparing the mice that have ischemia reperfusion with the scramble microRNA to the mice with the mimic of microRNA 486. And this slide on the left shows which genes are upregulated in the scramble treatment compared to the mimic and which genes are downregulated. And the bottom line of this slide is that we saw 551 genes or DEGs, differentially expressed genes, were affected in the proximal tubule compared to the scramble. And these genes included downregulation of classic AKI genes, including KIM1, NGAL, keratin 20, SOX9. And these have been described to be activated in ischemia reperfusion injury. With respect to the endothelium, it was a little bit more complex. There were 860 DEGs, and some of the classic injury models genes did not really pan out as being affected by the microRNA, although we did see that category of pro-inflammatory genes of a chemokine ligand family, CXCL11 and CXCL10, were downregulated by the microRNA. And I show this slide, it's another volcano plot, but it's just to highlight the impact of this microRNA on genes compared to sham mice. So on the left-hand side of the slide, it shows the comparison, and the take-home message here is that there are only about 184 genes that are differentially expressed with this microRNA compared to sham mice. When you look at the scramble microRNA compared to shams, on the other hand, there's more than 1,000 genes. So what this highlights is the fact that there is, the title of the slide is reversion, but I would say more of a preservation of the healthy transcriptome with use of this microRNA. So we went on to do some bioinformatics analysis of the genes which were affected by microRNA 486.5p and by the exosomes. And this slide shows the results of that using gene ontology and KEGG pathway analysis. And the pathways are ranked according to their p-value, the most significant to the less significant. So with respect to the microRNA, when we looked at the pathways that are upregulated, or the genes that are upregulated by the microRNA, there are only 2 pathways really, arachidonic acid metabolism and linoleic acid. But a larger number of pathways that are linked to the downregulated genes. So, and the most significant pathway was tumor necrosis factor signaling pathway, TNF. Highly significant that this pathway is affected by the microRNA, a number of other pathways affected. And these pathways are linked to biological processes, and I'm showing here the top 40 pathways. Response to cytokine is considered a biological process, which is heavily linked to TNF, of course. But we also saw within the top 40 pathways, many of the processes were linked to cell death or apoptosis. And again, I emphasize that these are the downregulated genes by microRNA 486.5p. So then we asked, well, what are the genes in this TNF pathway that are being affected, and what are the common genes for the microRNA 486.5p and the exosomes? There were 14 genes in total that were downregulated by microRNA 486.5p related to TNF, but 12 of these genes are in common with those genes which were also downregulated by the exosomes, and that's highlighted on this heat map. You can see that the profound lack of change of these genes compared to the ischemia reperfusion mice and compared to scramble. And this group of 12 genes contains some interesting genes, for example, endothelin-1, TNF receptor genes, VCAM1, and a number of chemokine genes as well as CSF1, which is macrophage colony stimulating factor. And we also looked at apoptotic genes. So which are the genes which are downregulated by the mimic and the exosomes that are in common? And we identified 49 downregulated genes involved in apoptosis with the mimic and the exosomes compared to ischemia reperfusion. Again, this is highlighted, and visually I think it's nice because you can see that these apoptotic genes are really not altered, or they don't really budge in terms of their expression levels compared to sham. So I'll just summarize what I've been talking about in this model. It's a model of ischemia reperfusion where we look at 24 hours after reperfusion, and then we've been comparing the mimic to the scrambled microRNA and exosomes. We were able to identify microRNA 486.5p in the kidneys by PCR and by ISH. And the microRNA potently protects against ischemic injury by all parameters tested, including apoptosis. And we were able to see, I didn't talk about this much, but decrease in PTEN, but an activation of pro-survival pathways. And most importantly, I think we have identified two categories of biological processes that are affected by this microRNA and by exosomes with common genes in both treatments. And I'll stop there, but I'd like to acknowledge the people in my lab and who've really done a lot of tremendous work in this area. Jose Vinas is a postdoc in my lab and was responsible for doing a lot of the experiments involving the administration of microRNA 486.5p. Matt Spence completed his master's degree in my lab, and he conducted a lot of the initial studies to look at the biodistribution of microRNA 486.5p and the differences between males and females in this model. Alex Goodsall is our resident pathologist at the KRC, does fantastic work with immunohistochemistry and immunofluorescence. We're really grateful to have him, and the ISH, which I showed you. Adriana Duvris, you all know, she's been working in the lab and contributed importantly to the isolation of proteins from endothelial cells and proximal tubules, which is a difficult process. Joe Zimpelman. Joe Zimpelman is a senior technician in my lab. He's been in my lab for more than 25 years now. I stole him from Steve Nadler's lab. I don't know if Steve actually remembers that, but Joe is just a fantastic person and one of the most, extremely high degree of integrity with the experiments that he conducts. Chris Porter from the OHRI was instrumental in helping us analyze the bioinformatics data, and Pearl Campbell from STEMCore assisted us greatly in creating the RNA libraries. Myra Trenton Sonoda is a research postdoc in my lab who assisted with the systematic review with regards to the studies on biological processes. And the systematic review was actually led by medical resident Sarah Zankar, and she spent a lot of work digging up the papers and analyzing them with us. And of course, Dave Allen helped greatly with the systematic review as well. And I have to give kudos to Rosendo Rodriguez from the Department of Medicine, who helped me try to understand meta-analysis and sort of the art of doing systematic review. And I'll acknowledge the funding bodies, and I'll stop there and be happy to take questions. Thanks
a lot, Kevin. That was really, well, really fascinating, actually, happening in our backyard here that we don't know about. I got a quick question. Maybe I missed it. The exosomes seem to be doing everything that almost everything that the mimic was doing. So why did you not just pursue that? Like, what's the rationale of looking at the mimic and some other delivery material?
Yeah, it's a great question, and the issue is that the exosomes contain other cargo, so we don't know what that cargo is doing. It can have off-target effects, and so there's multiple microRNAs that are present in the exosomes. There's also proteins and DNA, and then there's also the possibility of exosomes inducing immune responses with time. So we decided that to create a strategy where we could deliver it in an artificial environment, either a nanoparticle or a microvesicle that we create, and that's really the rationale for the direction.
Other questions for Kevin? Just yell out. I don't see the hands going up.
Hi, Kevin. It's Todd. That was amazing. Really, really enjoyable. Two quick questions. One kind of off topic, but could you envision miRNAs being used in kidney transplant for ischemia reperfusion directly kind of infused to a graft at the time of harvest? I'm sure that's been done. I don't know if you've got any kind of literature to support that. And I guess the second question is, and I may have missed this, but kind of the half-life of these miRNAs and also the kind of the duration of their effect. To me, this seems like a really cool technology, and that there's probably not any sort of long-term consequences or changing of gene expression over long periods of time with this. It seems to be quite short-acting, but has that been looked at before?
Yeah, so those are two great questions. With respect to transplant, yes, I would envision that's an obvious target for use of this kind of technology. And although we're not aware of any studies in humans, yes, in experimental models, microRNA has been delivered for delayed graft function and that type of thing in experimental models. And that would be, I think, one of the first areas for phase 1 studies to be done. And I should mention that Adriana is now conducting a systematic review of human studies involving MicroRNA and AKI. And so far, we have not come across any therapeutic studies or trials, but the majority of studies in humans have been directed at observational work in transplant patients and DGF. With respect to the duration of effect, it's actually an area of great interest for us that, and we're planning studies now to look at sort of long-term effects. What happens to these mice over time? Because there's some evidence that blocking PTEN actually can be detrimental with respect to AKI to CKD transition. And so we want to look longer out, and we also want to look at repeated dosing. Although we do see the microRNA at 24 hours within the kidneys, and it seems to be at fairly significant levels by ISH and PCR, we don't know how long that actually lasts and what the implications are. So many studies have looked at repeated delivery or repeated dosing, but we just don't have the data yet.
Are there other questions for Kevin? Nothing. We are after 9, so maybe we should conclude the round. So again, thank you everyone for participating. And again, thank you, Kevin, for really an excellent talk. And it's nice to see the work of our colleagues and where it's leading in their research. So thanks again, Kevin.
Thank you.